So this is embryology class and embryology which is systemic embryology. Have you already learned coelomic cavities? Development of coelomic cavity? Have you already learned development of foregut? Can't have a unanimous response. Okay, so that is also a part of systemic embryology. The 8 sessions that we had, they were of general embryology. The last session on general embryology was placenta, that was, we discussed it. And after that there was twinning and teratogenesis. Correct? So that was the completion of general embryology. And we started systemic embryology, and this is a part of systemic embryology. We are going to discuss development of the urinary system. Development of urinary system and reproductive system generally go hand in hand. So together they are called as urogenital system. There will be around 4 sessions on urogenital system, of which 2 we will be discussing urinary system and the remaining 2 the reproductive system, male and female. So what will be discussed is embryology of urinary system and congenital anomalies of the urinary system and their embryological basis over the next 2 sessions. Now when we learn systemic embryology, there are some principal heads under which we learn. Why we always classify, why in anatomy everything is classified? It is classified so that we can learn it better. So it's for our convenience that teachers, researchers, authors of the books, etc. classify things. So these 5 headings which you are seeing while we learn systemic embryology include the first and most important thing is germ layers of origin of that particular part. So when we say parts of urinary system include a pair of kidneys, a pair of ureters, unpaired urinary bladder and unpaired urethra. Then if you are learning development of kidneys, the first thing that you need to learn is which germ layers give rise to the kidneys. So germ layers of origin. And how many germ layers are there? 3. Name them. Endoderm, mesoderm and ectoderm. So which of the germ layers contribute to development of the organ? That's what is this. Next is primordial terms. Whenever we learn development of any organ, any part, there are certain terms which are used. For example, when we learn development of kidneys, we'll be learning something like pronephros, mesonephros, metanephros, ureteric bud, etc., etc. These are all called as primordial terms which are relevant to the development of that particular structure. Then, when does the development of that particular structure begin with respect to the time of the intrauterine life, time of development, and the size of the embryo? So you know what is the period of organogenesis? From which week to which week? Now that will depend on what is the definition of embryo, pre-embryo. We have discussed this the previous time. When does teratogenesis occur? First month. First trimester. That's even broader. First month or narrow tha. First trimester is even broader. 3 months ho gaya. Which week? 5th to 6th week. From the date of fertilization? Okay. What is the period of organogenesis? When all organs start developing? So you will say 3rd to 8th week, 4th to 8th week, 2nd to 8th week. So 4th to 8th week is a period of organogenesis when all organs start developing. And that is when, if teratogens are attacking the body, that's when the organogenesis itself is faulty. And that will lead to congenital anomalies. of different nature. So therefore, most of the organs, the beginning of the development will be in between 4 and 8 weeks. Most of them. Very few will be beyond that when they begin development. So this heading, blindly, if you write 4 to 8 week, very broad, you are right. You are not wrong. But what is expected is something very specific. Out of the 4 to 8 week, 4th week A, 5th week A, 6th week A, 7th week A, 8th week A. You will have to specify. Then, each organ will pass through certain stages of development. The first stage, followed by the next stage, followed by the next stage, and then the final organ. So that's called as stages in the development of the organ. And finally, we learn normal things so that we can understand what has gone wrong. So that's called as applied embryology in the form of anomalies of that organ. Got it? So these anomalies can happen if anything goes wrong in various stages of development. So these are broad principal headings under which we learn any organ development. If you remember this, you can easily draft an answer to a short answer question. Describe the development of kidneys. So these 5 heads should feature. And then your description of each head, supported by appropriate diagrams. Got it? The competency that you have to attain for this particular 2 sessions is describe the development of the urinary system. And then for these competencies, there are learning objectives that we will be able to achieve. So today, what was planned or what is still in plan is, you will be able to specify the germ layer that gives rise to the structural unit of kidneys and the ureters. List the primordial terms used to explain development of kidneys and ureters. Describe the stages in development of kidneys and ureters. Explain the process of ascent of kidneys. And if there is time, explain the embryological basis of congenital anomalies of kidneys and ureters. If not today, then it will be discussed on 30th of January. Got it? So germ layer of origin. You are very well aware there are 3 germ layers. Now tell me, which is the major germ layer that will contribute to development of urinary system? Mesoderm. Very good. Now the next question is, mesoderm is further classified. Can anyone volunteer, stand up and answer how mesoderm is classified? Yes, please. Paraxial, intermediate and lateral plate, which are components of which mesoderm? Intraembryonic mesoderm. So therefore, the first classification is mesoderm is classified as extraembryonic and intraembryonic. However, of course, when we say that there are 3 germ layers means they are going to give rise to the structure of embryo and its derivatives. So therefore, you are not wrong in saying endoderm, mesoderm and ectoderm, and mesoderm classification is also not wrong. It is always better to clarify, because extraembryonic mesoderm also exists as an entity. And intraembryonic mesoderm has got these 3 components. Now, out of these 3 components of intraembryonic mesoderm, which component is a forerunner for urinary system? Correct. So it's the intermediate mesoderm that gives rise to not only urinary but also genital. So urogenital system, germ layer of origin is mesoderm, and that too intermediate mesoderm. Okay. Now does it mean that only mesoderm contributes to development of the entire urinary system, or other germ layers also contribute? If the answer is yes, other germ layers contribute, then how does it contribute is the next question, for which I will ask you a question. What are the derivatives of ectoderm? Remember, this question I had asked you even in the previous session, and some of you listed a few structures. Correct. So nervous system is derived from ectoderm, and ectoderm, which is subdivided into surface ectoderm and neuroectoderm. Neuroectoderm is responsible for giving rise to various components of nervous system. Kidney as an organ, its coverings will also have some nerve supply. So therefore, nerve supply of the kidney will be derived from the ectoderm. But when we are talking of germ layer of origin, the principal structure or principal germ layer gives rise to the primordial structure of the organ. That is what we are concerned with. And therefore, the answer to this question is intermediate mesoderm. And just as our friend described intraembryonic mesoderm parts, paraxial mesoderm in the center, which is there around the notochord, followed by intermediate mesoderm, followed by lateral plate mesoderm, which is further divided into somatopleuric and splanchnopluric by formation of the intraembryonic coelom. And then of course they fuse, folding occurs, etc., etc. And then finally, in the intermediate mesoderm, the cells get reorganized to form what is called as nephrogenic cord, which is the first structure that will give rise to the future kidney. So nephrogenic cord further differentiates. Now let's come to the primordial terms that are used in development of the kidneys. Nephrotome. Nephro is for kidney. Tome is segment or section. Segment. Tome means to cut. So one segment of the developing kidney is called as nephrotome. Nephrogenic cord or ridge. Cord will be linear. Ridge also is linear but elevation. So in that intermediate mesoderm, there is an elevation that develops which is called as nephrogenic cord or ridge. Then pronephros, mesonephros, metanephros. As the name suggests, pro is one that comes first, meso in between, and meta that comes after. So pronephros, mesonephros and metanephros appear in that sequence. Then mesonephric tubules and duct. The duct, mesonephric duct, is also called as Wolffian duct. Then there is a term called as metanephric blastema. Then there is something called as ureteric diverticulum or ureteric bud. There is common excretory duct and angiogenic mesenchyme. So all these primordial terms should feature when you write an answer on development of kidneys. How? We'll just learn in the next few slides. So let's come to pronephros and its development. So once that nephrogenic cord or the ridge is formed in that mass of intermediate mesoderm, in the beginning of the 4th week, that is where the pronephro starts developing in the intermediate mesoderm, where in the cervical and upper thoracic regions of that developing small embryo. 7 to 8 tubules and one long duct. This is called as a pronephric or primary excretory duct that starts developing. So there will be one long duct like this and there will be tubules which will be entering into it, which are 7 to 8 in number. Finally what happens is tubules disappears, the duct communicates with endodermal cloaca, remember. Because finally kidney ureter has to go to the bladder and bladder is partly derived from cloaca. Pronephric duct, what happens to that is, it is annexed by the next developing kidney which is called as mesonephric kidney, and it receives a new name after that which is called as mesonephric duct. And such kind of pronephric kidneys persist in some fishes. It doesn't persist in human beings. But you know now that statement, ontogeny recapitulates phylogeny. That means all beginning development resemble in all organisms. And then, of course, depending on to which phylum the organism belongs to, the development changes and differentiates. So let's look at the pictures for pronephros. So these segments which are there, nephrotomes, then they become tubules. So these pronephric tubules will be there. This is the pronephric duct which is there. So they communicate with each other and then become vestigial. Then in the lower thoracic and lumbar region, the next thing start developing, that is mesonephric system. Is also shown here in segments. Another picture to show you the same pronephric tubules, then mesonephric tubules annexing to the duct, and finally the third one which will happen, we'll be discussing subsequently. Third picture. So 3 different books giving the same thing. So you refer to as many books, it will be the same that will be shown to you. Then finally pronephros disappears. The next kidney that will start developing is the mesonephros. And see the connection. This duct is opening into the cloaca, which is endodermal in origin. Now, what happens to these pronephric kidneys? There is something called as an internal glomerulus and external glomerulus that develops. And these are tufts of capillaries which develop from the angiogenic mesenchyme in that intermediate mesoderm. And they open into this pronephric tubules, which open into the pronephric duct. And this is connected to the coelomic cavity. So this is through what is called as peritoneal funnel. That means when this starts developing, the excretion happens just by filtration and it is given into the coelomic fluid. That's how it happens. Okay. But this system is not going to sustain long for such a complicated organism like the human being. So therefore, it disappears. So it is said that pronephros is nature's futile attempt to develop excretory system. But since ontogeny recapitulates phylogeny, it starts developing and then becomes vestigial. Then comes mesonephros. Mesonephros also starts developing in the fourth week, but at the end of the fourth week. That was beginning of fourth week, this is end of fourth week. The region of intermediate mesoderm where it starts developing is lower thoracic, upper lumbar regions. How many tubules start developing? 70 to 80 mesonephric tubules start developing, and one duct, which is the same, pronephric duct continues down, now receives a new name. That's called as mesonephric duct or Wolffian duct. Out of the 70 to 80 tubules, the proximal ones disappear between 5th to 8th weeks. Only 26 distal tubules and the mesonephric duct persist. This duct communicates with the endodermal cloaca, and this system persists in most fishes and amphibians. Now, as the mesonephros starts developing, there are two ridges that form. The mesonephric ridge of intermediate mesoderm, and medial to it, there is another ridge that simultaneously forms, that is called as gonadal ridge, which will be the forerunner for the development of gonads, either ovary or testis, depending on what is the chromosomal complement of that embryo. So let's look at it pictorially. So this is where the mesonephric duct is. One mesonephric tubule is opening into the mesonephric duct, and the mesonephric tubule is going to reorganize. Then angiogenic mesenchyme comes and forms the tuft of capillaries here, that is glomerulus, and all those things will happen. So this is one segment, that is horizontal section passing through the, and this is viewing the posterior wall of the developing intermediate mesoderm. So these are mesonephric tubules opening into this mesonephric duct, which is opening into endodermal cloaca. This is a transverse section to show the same. This is the mesonephric duct. The nephrogenic cord and the tubules in the lower thoracic upper lumbar region start developing into the tubules of kidney. This is the mesonephric vesicle from one tubule, and this is mesonephric duct. So they will develop communication. And this ridge will differentiate into the nephrogenic ridge, the nephrogenic, the ridge and the gonadal ridge will develop medial to it. So that's why this initial ridge is called as urogenital ridge combined. And then it will split into two, urinary and genital or gonadal. And this vesicle now becomes a tubule and joins with the duct. And then it coils. And as it coils from the dorsal aorta, which is developing here, again the angiogenic mesenchymes give these glomerular vessels, then the capillaries develop, and finally glomerulus. And then the complicated structure also develops. Now all this is happening, and simultaneously you see the gonadal ridge has started differentiating into the primordium of the gonad here. But mesonephric kidney is formed. It is still not able to sustain, because the glomerular system is formed, filtration happens, but the differentiation of mesonephric tubules into the loop of Henle doesn't happen. So therefore, the two functions of the adult kidney, which is apart from glomerular ultrafiltration, selective reabsorption and tubular secretion, do not happen in the mesonephric kidneys. So therefore, it is nature's futile attempt, not futile, misguided attempt. It tried to evolve, but it becomes misguided. It couldn't sustain. So it's a misguided attempt to form the excretory system, and therefore what is required is metanephros. So this is the story how the development of kidney evolves. So 5th week, when the size of the embryo is 5 millimeter and the crown drum length is 5 millimeter of the embryo. Same intermediate mesoderm is responsible. The sacral region of the developing embryo where it develops, it forms kidneys in reptiles, birds and mammals. Humans are mammals. Metanephros, not only metanephros, but there is something called as collecting part and excretory part of the kidney. You have learned gross anatomy of the kidney. When you learn gross anatomy of the kidney, on the surface you learned anterior surface, posterior surface, upper pole, lower pole, medial border and lateral border. When we take a longitudinal section and open and see the section of the kidney, have you seen that? So when we see that, what we see is outer cortex, inner medulla. Inner medulla is pyramidal in shape. And in the pyramid, apex of the pyramid opens the minor calyces, which unite to form the major calyx. Major calyces unite to form the renal pelvis. So all this is called as the collecting part of the kidney. And what is lying inside? Nephron, the ultrastructure. But nephron has got excretory part, because that is where glomerular filtration will happen, then tubular secretion and selective reabsorption will happen. So till the part which is called as collecting tubule, till that part is called as excretory part. From collecting duct onward, collecting tubule onwards, becoming collecting duct, minor calyces, major calyces and pelvis, renal pelvis, it's all called as the collecting part. It is collecting. Excretory part is the one which forms urine. Collecting part is only a channel for conduction of this urine. That's the meaning of the thing. Okay. Collecting part develops from what is called as ureteric diverticulum, which develops from dorsomedial side of the mesonephric duct. It's also called as common excretory duct. The excretory part develops from metanephric blastema. So when you are asked in the exam, list the two sources of development of kidneys, the answer is collecting part, excretory part. Collecting part develops from ureteric diverticulum from mesonephric duct, and excretory part develops from metanephric blastema. That's the answer. Division of the ureteric diverticulum within metanephric blastema occurs to form the pelvic calyceal system up to collecting tubule, and angiogenic mesenchymes form the glomerular plexus, endothelial and mesangial cells which are there within the kidney, details of which you will learn when microanatomy of urinary system you will be. Okay. We stop here. We give you time to submit journals. Come back, go to washroom, come back, sit here. Community medicine class will occur. When that class gets over, and if time remains, we'll continue our discussion.